Hi, welcome. This is the Chapter 11 review. Chapter 11 is the cell cycle and cell division. This first review, we're going to look at the cell cycle specifically. Right, now, the cell cycle, the big picture we're looking at here is cell division, and different types of organisms use cell division for different functions. So unicellular organisms, or organisms made of one cell, they use cell division to reproduce. So they'll go from one cell, and to make more of themselves, they just divide in two, and now you have two new cells. So if you look at this picture at the bottom left, these are yeast cells, which are single-celled fungi, reproducing. They do so just by budding, which is splitting off from one cell, making new cells. And they're identical to each other. Now, multicellular organisms use cell division for other things. So they'll use it for growth and repair, and also regeneration. Okay, so if you think of your skin, if it gets cut, it'll heal. Your skin cells are dividing, and they'll go through types of division to repair it. Or as an organism grows from a child to an adult, it gets bigger, and it's because the cells are dividing. That's growth. And some organisms can regenerate tissue that's lost, like this lizard can lose its tail as a defense and then grow it back. Now let's look at prokaryotic cell division. Remember prokaryotes, things like bacteria, single-celled, single chromosome, no membrane-bound organelles, no nucleus. In order for one of these single-celled organisms, like a bacterium, to divide, there's general signals that occur, and they kind of make sense if you think about it. Okay, so the first signal is some sort of signal to reproduce or begin the cell division. Now it's going to divide, so the first thing it has to do then is replicate its DNA, its genetic information, make a complete copy of it so each new cell gets a copy. You then segregate that DNA to the two new cells are going to form, and then you have cytokinesis, which is the splitting of the cells in two. So this process in prokaryotes, like bacteria, is called binary fission. Binary means two, and fission is to separate. So you take one bacterial cell at the top, and through this process, we have binary fission, and we create two new cells, which are essentially identical copies to each other. So inside this bacterial cell, there's one single circular chromosome, right? And it replicates from its origin of replication and makes a copy of that single chromosome. That chromosome is then divided or segregated into two opposite sides. And then the end result, after cytokinesis, we have two new daughter cells that are identical copies of the original one. Okay, this is identical copies. Now in eukaryotes, organisms with a nucleus, there's different types of cell division. There's mitosis, which is more specifically the nuclear division, and then when this whole cell splits into that cytokinesis. But mitosis is used for growth, repair, and regeneration. Okay, you start with one cell, you go through one round of cell division, or one nuclear division and cell division, and you end with two daughter cells, and they're identical genetically to the cell that you started with at the top. So if you look at the diagram, one cell goes through one round of division, we end with two daughter cells that are 100% identical to the parent cell. Okay, so that's for growth, repair, and regeneration. That makes sense. So this would be something like your skin. Again, if it's skin cells going to divide to make new skin cells, you would want the new cells to be 100% identical to the one they started with, right? Now, meiosis is a type of cell division that's used for reproduction. So this is the bottom figure here. And what happens during meiosis is you start with one cell, and we go through two rounds of cell division this time, and we end with a total of 4 daughter cells, okay, on the bottom. And these 4 daughter cells have half as much genetic information as the parent cell, and the genetic information also has other variations. So they're not identical to the cell they start with, and meiosis is how we produce our gametes. Okay, and it's very important that we create variation in our gametes for sexual reproduction, and we'll talk about that later in meiosis. Okay, now let's look at the cell cycle as a whole process, and this is dealing with eukaryotic cells. Okay, basically the eukaryotic cell cycle can be broken down into two phases. Interphase, which is a time when the cell is kind of being a cell or preparing for cell division. And then the other phase is the M phase, or mitosis and cytokinesis. Mitosis refers to the nuclear division, and then cytokinesis is when the cell actually divides. So let's see a figure to help put this into perspective. So this figure here is illustrating the cell cycle. All right. All the area in brown here, this is interphase. Okay. So this is the majority of the cell cycle. And during interphase, there's subphases, right? There's a G1 phase, or gap 1. During this phase, the cell is doing what that type of cell does. So it's being a skin cell, or it's being a bone cell, or a muscle cell. And during this phase, another thing it does is it starts to prepare for the next phase, S phase, okay, which is DNA synthesis phase. So during S phase, the cell copies all of its genetic information. Okay, that's an important step. If the cell is going to eventually divide at some point, it's going to need to pass a copy on. So S phase stands for synthesis, or DNA synthesis, and we make a copy of all the genetic information. G2 phase is a preparation for mitosis. Okay, and so then that brings us to the blue region here, which is mitosis, which is a nuclear division. So you see you have one nuclei, we split it into two, and then cytokinesis will split these cells into two. All right, let's look at that cell cycle a little more. We mentioned that the interphase is the brown region of this cycle. We have G1, S, and G2. So let's look at what happens to the genetic information. So from G1, we enter S phase. During S phase, we make a copy of all the genetic information. So we could say that during S phase, DNA replication occurs. Okay, each chromosome is replicated and joined, forming what we call sister chromatids. So here's an example. If we look at the drawing I have prepared here, during G1 phase, you have each one of our cells, we have two copies of all our chromosomes. So we have one chromosome 1 we got from mom, one we got from dad. So we have two chromosome 1s. We also have two chromosome 2s, illustrated in red. We'd also have two chromosome 3s, two chromosome 4s, and so on. So as we go from G1, and once we enter S phase, we copy all the information we have. We copy our DNA. We go through DNA replication. So chromosome 1 makes a copy of itself, and we get this kind of X shape for a chromosome that you're familiar with. Okay, both of our chromosome 1s do that. Same with our chromosomes 2. They're now duplicated compared to what they looked like during G1 phase. All right, so now we have for chromosome 2, this top one, we have two sister chromatids, right, of chromosome 2. All right, so we can say that during S phase, we have replicated all our genetic information. And this will be important for later when we go through the actual nuclear division. We want to divide this genetic information into the two nuclei. So here's a picture of the cell cycle. And one thing that's important about the cell cycle is that it's very highly regulated. We don't want our cells to divide when they shouldn't divide. So we're going to talk briefly about how our cell knows when to go from G phase to S phase, and then from S phase to G2 phase. This is very highly regulated because when our cells start to divide when they shouldn't, we get tumor formations. So if you look at this illustration on the bottom, we have some skin cells, and we have the beginning of this mass of cells that is growing uncontrollably, and this is a tumor. Okay, the cell's going through this cell cycle even though it's not supposed to. All right. So how do we control cell division, the cell cycle? Well, there's enzymes called cyclin-dependent kinases. We know they're enzymes, they end in ASE. That's a good indication that it's an enzyme. We also know something about kinase. Remember, a kinase is an enzyme that adds a phosphate to something, and that process is called phosphorylation. All right. Now this name, cyclin-dependent kinases, we abbreviate it CDK. So cyclin-dependent kinases, they're enzymes or kinases that depend on cyclin. Okay, so let's look at this illustration. Here's the enzyme CDK. If we look at the enzyme, in this area right here is its active site. Okay, it's actually closed right now. So when cyclin-dependent kinase CDK, it has to bind to cyclin in order to become active. So here's the active site. It's become available. All right. So in order for this enzyme to be active, in order for CDK to be active, it has to bind to cyclin. And that makes sense because the enzyme is called a cyclin-dependent enzyme, so it depends on binding to cyclin to be active. This is called allosteric regulation. When something binds to an enzyme and locks it on the on position, that's that type of allosteric regulation we learned about in Chapter 8. So now this cyclin-dependent kinase cyclin complex we see right here, part B of this diagram, is activated. So now the enzyme is going to do what it does. It's going to bind to a substrate, a protein, and it's going to add a phosphate to it. it gets a phosphate from ATP. It takes one of those phosphates, adds it to this protein. As a result, that ATP becomes ADP, right? And this protein down here in the bottom left gets phosphorylated. And that's going to modify the function of this protein. This protein is called RB protein for retinoblastoma. And when it becomes phosphorylated, it's inactivated. So what does that mean? Let's take a look at the cell cycle to see the consequence of this. So here's a cell cycle, and there's a major checkpoint, a restriction site right here where this R is. Okay, and the cell's not going to go from G1 to S phase unless it passes through this point. All right. Now what's preventing the cell from going from G1 to S phase right now is this RB protein we see at the top. So what happens is cyclin binds to CDK. Okay, it activates this enzyme because CDK is a cyclin-dependent kinase. It's dependent on binding to cyclin. When it does, the active site opens up. So this enzyme complex can now bind to retinoblastoma protein. Okay, that fits right into this active site. And when it does, the RB protein becomes phosphorylated. And when it's phosphorylated, this protein is inactivated. So now that it's inactivated, the cell's free to go on to the next phase of the cell cycle. Okay, so that's a very important regulation checkpoint of the cell cycle. Now it turns out if we look at the cell cycle in this diagram, you'll see there's several different types of cyclin CDK complexes that regulate different stages. So there's one in G1 we saw, there's one in the S phase, there's also one in late G2 phase, and there's even one through the mitosis phase. And each of these are checkpoints that allow the cell to determine if it should move on to the next phase of the cell cycle. There's lots of reasons why you'd want to kind of have regulation at these steps. These cells stop at these points and they take a survey of their DNA and they see if it's damaged. If it's damaged, they can pause and they can repair it. Because you don't want damaged cells to divide, so you need a moment to repair them. And if there's too much damage, the cells will actually go through a process called programmed cell death, which we'll talk about later. But the idea here is you have a damaged cell, you want to take out that one cell before it divides and becomes 2 damaged cells, which will divide and become 4 damaged cells. So we have all these checkpoints to make sure we don't let these cells get by. And when they do, that's when we have serious problems like tumors and cancer. Now cyclins are so important because they help monitor the cell cycle or regulate it. And as you can imagine, then these cyclins themselves are also regulated. So what we're seeing in this illustration here is that the cyclins are what we call transient. That means they're synthesized and broken down as needed. And so the cyclins required for the G1 to S transition are synthesized during G1 phase. Okay, they formulate the cyclin complex and then they're degraded, okay, after the cell passes through that checkpoint. So that's the overview of the cell cycle. In the checkpoints and the regulation. The next review we'll look at the mitosis.